Okay, I think we're ready to start. I'm David Melson. I'm the one that's been bombarding you with emails. You're probably sick of the emails and sick of me, but it's worked because we have a good audience here, and I know that a lot of people have registered to watch online. I'd now like to ask Dr. Lindsley Bilsland from the Parkinson's UK, the new Managing Director of the Parkinson's Virtual Biotech, to give you a proper welcome. Lindsay.
Good evening and welcome to the 14th Annual Edinburgh Parkinson's Research Lecture. Thank you all very much for being here this evening. As David said, my name is Lindsay Bilsland and I'm the new Managing Director of the Parkinson's Virtual Biotech. And I'm delighted to be here this evening, so thank you to David, who's the chair of the Edinburgh Parkinson's Research Interest Group, as well as Stephen Brannan, the chair of the Parkinson's UK Edinburgh branch, for inviting me here. So the history of this lecture is quite remarkable. Started back in 2012 by Professor Ken Bowler and the Parkinson's UK Edinburgh branch for the local Parkinson's community. And it's grown ever since. So as I said, this is the 14th lecture, and there are several hundred people here in the room, many more online. We have a real diversity of attendees, from people with Parkinson's and their families and carers, through to healthcare professionals, researchers and students. And then tonight we're also joined by the executive leadership team from Parkinson's UK. So tonight the focus of the lecture is on stem cell therapies for Parkinson's, and you may know that there are some stem cell therapies in the clinic at the moment, and I'm sure we'll hear more about that later on. But I wanted to highlight the important contribution that Parkinson's UK played in stem cell therapy, in that we funded some of the early formative work that led to increased understanding in how stem cells could actually be used as a direct treatment for Parkinson's, as well as how stem cells could be used as a tool to support the testing and the development of other treatments. We also funded, back in the 1990s, we funded some early transplantation studies. Now these were done in a small number of people and the results were inconsistent, but for a small number of people with Parkinson's, they experienced quite significant improvements. And these studies were the forerunners to the clinical trials that are ongoing today. So I'm really looking forward to hearing an update later. Now, this stem cell research is part of a wider research portfolio funded by Parkinson's UK. So since 1969, Parkinson's UK have provided 130 million in funding for research. And that includes 15.6 million that was awarded in 2024 alone, making us the leading European funder of Parkinson's research. And we fund research from cause to cure. So that could be research into identifying mechanisms that cause Parkinson's, through to funding preclinical and clinical development of new treatments. They may be both drug and non-drug approaches, and maybe approaches that have the potential to stop or slow progression of Parkinson's, as well as those designed to help treat the symptoms. We also provide funding for infrastructure such as the Parkinson's Brain Bank and the Innovative Trials Platform. And the Parkinson's community is the driving force behind our research. So it's built on the priorities of people with Parkinson's and shaped by them. So, for example, we have people with Parkinson's on our funding committees. So I joined Parkinson's UK about 2 months ago now, and I lead the Virtual Biotech. And the Virtual Biotech can be considered as the global drug discovery and development arm of Parkinson's UK. And so our goal is to support the development of new drugs for the treatment of Parkinson's. And these may be disease-modifying or symptomatic approaches. And we provide funding to both academics and biotech companies to develop new treatments, whether they're in the preclinical space, so that's the kind of lighter blue. So this could be when you're testing new drugs in a dish full of cells or in an animal model, through to clinical trials in the darker blue, where you're actually testing a new drug in a person with Parkinson's to see if it's effective. And we provide not only funding, we also provide access to our Parkinson's network, and we also provide internal expertise. So we have an internal team that has that detailed understanding of the biology, of the therapeutic landscape, and of the challenges in developing drugs for the central nervous system. We collaborate with external experts in the Parkinson's field, and we also work really closely with people with Parkinson's to make sure what we're actually doing is relevant. And then finally, we have established relationships with industry partners, with investors, and with other funders such as the Parkinson's Foundation to help build that supportive ecosystem and accelerate development of new treatments to get them into the hands of people with Parkinson's as quickly as possible. So we have 11 active projects in our portfolio, shown by the names kind of above and below the line, and we're looking to build out this portfolio and diversify the projects within it in order to maximize our chances of success. So now, let's hear more about stem cell therapy. So I'm going to hand over to Professor Thilo Kunath from the University of Edinburgh, who's going to introduce our speaker tonight, Dr. Kirkeby. But thank you all very much, and I hope you enjoy the evening.
Thank you, Lindsay. So welcome, everyone. And I just want to, before I start, is to reiterate the point about Parkinson's UK funding. It's been transformational for so many areas within the UK for people with Parkinson's, but also including myself, right? I mentioned earlier today how the Parkinson's UK Fellowship transformed my whole trajectory in my career, so thank you very much. So it's my huge pleasure to be introducing Agnete Kirkeby today, our 2025 Edinburgh Parkinson's Lecturer, which is now in its 14th year. So Agnete did her PhD at the University of Copenhagen in 2009, finished, so I think that makes her the youngest Edinburgh Parkinson's lecturer that we've had so far. She moved briefly to Lorenz Suter's lab in New York to do a postdoc, and then to Malin Parmer's lab in Lund to do a postdoc, and published a very seminal paper in 2012 that described how you can turn stem cells into dopamine-producing neurons. And I checked online today, this paper has been cited over 800 times, including several times by me, but many other people. So this is a real key finding, the basic property of turning a stem cell into a neuron. She set up her labs, I should say, 2015, 2017, in Lund and in Copenhagen. And if you know the geography, they're quite close to each other across the bridge. But as of 2 days ago, she is fully based in Copenhagen at the Novo Nordisk Foundation Center for Stem Cell Medicine. So she's going to tell you today about a clinical trial that Malin Parmer, Agnete, and Roger Barker have started called STEM-PD. So I think this is one of the exciting high points of today. I wanted to say just one more thing. So Agnete is an exceptional basic scientist. So she's not a clinician. Is that correct? No? Right? She publishes really beautiful work, I think, but what's impressive about Agnete, and really impresses me, is that she is extremely determined, extremely persevering, and she does all the regulatory paperwork, all the hard work of bringing a basic therapy to a clinical trial. And that is less basic science and less fun, but she's done it, you know. So I'm just super impressed and super proud. So I'm pleased to introduce you to the 14th Edinburgh Parkinson's lecturer, Agnete Kirkeby.
Thank you, Thilo, for those very kind words. Thank you for the invitation, Thilo, David, Parkinson's UK. I'm very honored to be here. Thank you to all of you for coming and for all of you online. I'm humbled to be part of such a magnificent lineup of speakers. As Thilo mentioned, I am not a clinician myself. I'm a basic scientist, but I'm going to tell you today about the journey that we've gone through of taking a discovery we made in the lab to take it all the way to the clinic, to do clinical trial in Parkinson's disease patients, because we wanted this to really be something that could help individuals living with Parkinson's disease and not just our rats in the lab. So I'll take you through this journey, and then in the end I'll give you an overview of what other people in the field are doing, because we are not the only ones working on a stem cell therapy for Parkinson's disease. So you'll get a bit of an overview of how things are standing today. I'm not going to tell you about how it is to live with Parkinson's, because I know that many of you in the room know much more about that than I do. I will just briefly say, as many of you already know, one of the key hallmarks of Parkinson's disease is this loss of dopamine neurons in the substantia nigra. And we've known for a while now that the loss of this particular subtype, although it is not the only subtype that can be lost in the brain, but the loss of this particular subtype, and it's actually a very small population of neurons lying very deeply in our brains in the substantia nigra. And when we lose these particular neurons, it dramatically affects our motoric function. And this is what we see in Parkinson's disease. It is the loss of these dopamine neurons that in particular cause the motoric symptoms of the disease. So put very simply, the idea, which was actually stated in Lund already back in the 70s, they were working with rat models, was to see if it might be possible to somehow get new neurons that could be put into the brains of individuals with Parkinson's to regain this function of the dopamine neuron in the brain. So that sounds like a very simple concept. It's not that easy in practice. So this was an idea that really came to life. I told you already in the 70s, actually, they started working with rats. At that time, we did not have stem cell technology. It wasn't really, we didn't even know what stem cells were at that time. I wasn't even born. But what they did at that time instead was they investigated where these neurons were coming from in the embryo. And they found out that the dopamine neurons are derived from a very particular region of the developing fetal brain, a region called the midbrain. And then they started taking fetal tissue from rats and they put it in to aged rats, and they could see that they could actually get these neurons to survive after transplanting them into the brain. So they thought, okay, let's try to do this in humans. And they were able to get donated fetal brain tissue from legal abortions. This was tissue that was anyway going to be discarded, put into the trash bin. So they were able to get this tissue donated. They could then subdissect out this very small region of the brain and make a cell suspension, and then they could transplant this into the brains of individuals with Parkinson's disease. And I'm going to show you now an example of one person that was transplanted in Lund. This was actually not a classical Parkinson's disease patient. This is an MPTP. This was an MPTP intoxication. So this is an individual that had lost the majority of her dopamine neurons through intoxication with MPTP. She was a drug addict, and the MPTP intoxication came from a contaminated batch of heroin. So this is an individual who really had very severe dopamine loss. And I'm going to show you here what this looks like. It looks like I've put the video on slow motion, but I have not. This is real time. She's asked to tap her fingers, and then she's asked to stand up. And as you can maybe see, this is very difficult for her. This is now the same individual after transplantation with fetal-derived dopamine neurons. She is off any type of medication in these videos, so what you're seeing here is what you would call in the off state. There's no L-Dopa or other dopamine-modifying medications. What we're seeing here is really an effect of the transplant that she received, the dopamine neurons that were put into her brain coming from these fetal brain tissues. So she's one example, and a very striking example of how this can work if we do it right. If we transplant enough dopamine neurons into the brain, if they survive and they stay there, then they can actually have a very prolonged effect. And we've seen in one post-mortem brain, 24 years old graft from an individual that was transplanted with fetal tissue. You can see here how even 24 years after transplantation, the new dopamine neurons look healthy. They are thriving in the brain. They look exactly like we would want a healthy dopamine neuron to look like. So this really tells us that when you have these neurons in the brain, they can survive for many years in the brain of the patient. And it's important to say here that we know also from these individuals that you don't need lifelong immunosuppression, such as you would normally need, for instance, for a kidney transplant or a heart transplant or the like. So there's something special about the brain, which means that we can do just a short term of immunosuppression, and then the graft will still stay for many years. This is data from 2 individuals that were transplanted in Lund. And these were individuals that responded very well to the transplant. You can see here both of them went off their dopaminergic medication, and they actually went off their dopaminergic medication for more than 10 years because the graft was really working for a prolonged amount of time in their brains. So these are examples of how this can really work very well when the transplant is surviving and functioning in the brain. There has been a follow-up, a newer study in Cambridge and Lund from 2015 to 2023. This was led by Roger Barker from Cambridge. And here they transplanted 11 individuals with fetal tissue to try to see if they could standardize the procedure, to see if they could optimize it to make it more less variable, because there was variability between the patients. Unfortunately, what they saw was that most of the individuals just had not received enough tissue. There wasn't enough dopamine neurons in these transplants, so there was a lot of variability again. There were, again, as there had been before, individuals that responded very well. Here is participant number 60. This is a PET scan before transplantation and 18 months post-transplantation. And what you're seeing here is really a very significant PET signal in the transplanted brain. And this individual actually decreased in the UPDRS score in off with more than 20 points. So this is very significant. But there was a huge amount of variation, as there had also been in the previous trials. So what have we learned from these cases? Most of them have not been very well controlled. Each individual has received a different amount of tissue, different quality of the tissues, very little standardization between these cases. We have learned that the transplanted dopamine neurons can survive for more than 20 years in the Parkinsonian brain, even after only short immunosuppression. We've also learned that it's a slow effect. So I showed you the videos 5 years after transplantation, right? And that's because the effect of the graft really takes time to incorporate into the brain. You can imagine it's a new neuron you put in, it needs to mature, send out its axons and dendrites, it needs to contact the host brain, and only when that contact is fully established will the transplant work fully. And that can take actually up to 3, in some patients even up to 5 years for the maximal effect. And then this problem, it was highly variable. As Lindsay also told you, it was variable and it was something that could not be scaled up to many patients. In the meantime, as these trials were ongoing, we got access to stem cell technology. And what does this mean? Well, this means that this gave us the possibility to investigate if maybe instead of using this very variable donated fetal tissue, there was never enough, there's always a scarcity, well, maybe instead we could manufacture dopamine neurons in the lab and then transplant these to the brains of individuals. And this would make a huge difference because there was always a problem with the fetal tissue, and often there was not enough because you actually need between 3 to 4 donated fetal brains to have enough to just transplant one side of an adult human brain. So there was never enough. But with stem cells, potentially, you could generate indefinite amount of cells. So what is a stem cell and where do you get stem cells from? And actually, we do have stem cells in our bodies. We have stem cells in our skin, in our blood, muscle, intestines. These are stem cells that are active every single day in our bodies. But these are what we call tissue stem cells. They are designed to only make the tissue that they reside in. So a skin stem cell is very good at making skin. You can see this if you get a wound or a burn. Very quickly, the skin stem cells will come in and they'll heal the wound. And the same for the intestine. It constantly regenerates your intestinal epithelial layer, your muscles if they get injured, and your blood constantly regenerates. But they cannot make other tissues. A blood stem cell makes blood. A skin stem cell makes skin. None of them can make brain. And this is the problem, because in the brain, we don't really have a good population of stem cells. There is no normal regeneration, no normal formation of neurons in the brain. So what do we do for these diseases where it's brain cells that we're losing? We don't have any stem cells in our body. So this is where we have to go back in development. We have to, so to say, rewind time in development and actually go all the way back to right after the egg was fertilized by the sperm cell, the earliest stages of our development in the womb, around 4 days after egg fertilization. At this stage in our development, which we have all gone through, but which lasted only around 24 to 48 hours, we have this structure called a blastocyst. So it only exists for a few days, and then it disappears again. But what's so magnificent about the blastocyst is that in the middle here of the blastocyst, which is a tiny, tiny structure, we have what we call pluripotent stem cells. And the pluripotent stem cells are the cells which will eventually give rise to the entire human body. This little clump of cells in the very early time point in development will form all the organs of our bodies. And this is why we call them pluripotent. It means they have the capacity, the potency to form every single cell in our body. They are the only stem cell that can do this, and they only exist in this super brief time point during development, and then they disappear. So how do we get access to these? Well, this was actually really a breakthrough in biotechnological research. You can see here how small the blastocyst is. It's a tiny, tiny ball of cells. In 1998, it was discovered in the US how you could take out these cells from the blastocyst and you could grow them in the dish. And now they start dividing and forming copies of themselves if you do this the right way. And they can actually divide forever and ever. They will form an indefinite amount of copies of themselves, which is a really cool thing because this means that actually you can, from one single fertilized egg, you can form stem cells for the entire world population. I'm not exaggerating, that is actually true. They will not stop dividing, which is an amazing feature, right? This means now we can make indefinite amount of cells. So not only will they continue to divide, we can then also freeze them down, we can share them with researchers all over the world, and we do this. We work with cell lines that have been derived all over the world, some of them many, many years ago, and they're still perfectly fine to work with. And then the cool part, they divide and divide, but then they can also make all these tissues of the human body, every single cell type of our human body, including the cells that are very difficult to get access to otherwise, like the neurons of our brains. And this is one thing that we as stem cell scientists have worked on really intensely, is to understand how do we control to get these stem cells to produce just the cell types that we're interested in. And now through many years of research, groups all over the world have been able to make we call them protocols, kind of like recipes to make different cell types from this pluripotent stem cells. For instance, we can make now insulin-producing pancreatic cells, three-dimensional human intestinal tissue that looks just like a human intestine, three-dimensional human kidney tissue that can even filtrate like a human kidney can do it, a human eye cup that has the exact same size as a human eye cup, and even a small beating human fetal heart. All of this produced in the dish from stem cells. That's pretty amazing. And of course, this stem cell technology now also allowed us to produce neurons. And how do we now make the right neurons for Parkinson's disease? Now, the real challenge here is that you should know our adult brains are super, super complex, right? It's not just a ball of neurons. You've probably seen a brain. It has very many different regions, and each of the regions of the brain have super specialized neurons that can do one specific, it can fulfill one specific function. We actually have more than 1,000 different subtypes of neurons in our brain, each of which has a super specialized function. So when we want to replace a neuron in the brain of an adult human, it's very important that we make the right type, because otherwise we're just going to mess up things. So one thing we have to understand as stem cell scientists is, where do the dopamine neurons come from? Because this is what we want to generate for Parkinson's. And here, a lot of research, in particular also from Lund, but also other places, have found that the dopamine neurons were derived here from the ventral midbrain of the embryo, and that this is really the region where they are born. So the job for us as stem cell scientists is to take these pluripotent stem cells and to add certain signals, we call them growth factors, to the dish, which will now coax the stem cells to become midbrain cells. We need to kind of fool them to think that they are here in the embryo, so they will form this region of the embryo. That's our job. And that's not very easy. In our case, we spent around 5 years to figure out the factors that needed to be added to generate exactly this region of the brain. It's a combination of 8 factors. It takes around 16 days, but every single factor is super important. Thilo would know this. He does this also in his lab. If you leave out just one of them, or if you add the 10% wrong concentration of one of them, you get the wrong cells. You will not get the dopamine cells. So this is really the trick. So then you can take these cells and you can mature them in the dish, and then they become mature neurons. So cells that can really form these long axonal fibers, they start becoming electrophysiological active. They look like a dopamine neuron should look. But in fact, we're not so interested in that. Instead, we want to know, can we take these midbrain cells, we call them progenitors, they're kind of teenager cells, they're not really adult yet, they're young, and they're not really yet a final dopamine neuron, but they will become a dopamine neuron. So we want to see, can we take these young teenager cells, can we put them into the brain of an animal model, and can they then mature and become active dopamine neurons in the brain? And what we use here is a rat model where we first take away all the dopamine neurons on one side of the brain. And you do this by injecting a toxin on one side. We could inject, for instance, MPTP. We use another toxin that's called 6-hydroxydopamine. And that now removes all the dopamine neurons on one side. Then on the other side, you have an intact system of dopamine neurons. This is a staining here for dopamine neurons in a rat brain. So dopamine on one side, nothing on the other side. And now you can put the stem cells into the lesion side through transplantation. And then you can check if your stem cells are also expressing the markers of a dopamine neuron. And this is now 2 brains, of which one was transplanted with cells in which one of the 8 factors was left out of the recipe. And then you get the wrong type of cells. They don't express dopamine. Whereas if you do the recipe correctly, you get the dopamine cells. And you can check now if they're functional. And you do this by giving the rat amphetamine. And what happens is when you give a rat amphetamine, you get a lot of dopamine release on one side of the brain and nothing on the other side because we lesioned it. And what then happens in a rat that has gotten amphetamine like this is it starts rotating around itself because it now has an asymmetry of dopamine in the brain. A lot on one side and nothing on the other side. It starts rotating, and actually, that can be a PhD project in my lab. Sit and count how many times does the rat rotate around itself. That is a core assay we use a lot. This is what we call the rotational asymmetry test. And now we can test, okay, what if we give it transplant of stem cells on the lesion site, because now we would expect this side to also produce dopamine. So the asymmetry of dopamine should be evened out. And that's exactly what happens. This is a graph showing you how many times the rat rotates around itself. And if you give it the wrong type of stem cells, it will continue to rotate around itself. If you give it the right dopamine stem cells, it will decrease. And even sometimes you can get it to rotate the other direction, because the stem cells can actually be more efficient than the rat's own dopamine neurons in producing dopamine. And of course, this is not really an animal model of Parkinson's, but it is an animal model of dopamine function. And it's very useful for us because it tells us that the transplanted dopamine neurons are surviving, they are integrating, they're communicating with the host brain, and they are producing dopamine, which is what they should. We've done tests also in the pig. This is an MPTP lesion pig. We did this, collaborated with another group in Denmark. And what is cool about the pig is it fits right into a PET scanner, which is the same type of PET scanner that we actually use for humans. So that means you can look at the transplanted cells in the pig while it's still alive. And that's what we're doing here. These are two PET tracers, which some of you might be familiar with. They're very commonly used in Parkinson's research. And they both mark the signal from functional dopaminergic terminals. And what you can see here is that in the transplanted side of this pig, there is a very clear signal from the dopamine neurons that have been transplanted. And these are, again, derived from the stem cells produced entirely in the lab. So what these two animal models tell us is that the dopamine neurons can do their main job, right, which is produce dopamine. That's what we want them to do, right? That they can definitely do. But there is another very important job of a dopamine neuron, and that is to clean up after itself. Because once it's no longer a teenager and it's become an adult dopamine neuron, it learns to clean up after itself. And the way it does this is because it expresses what we call the dopamine transporter. And the dopamine transporter has this cool function that it can take up dopamine if there's too much, put it back into the cell. And only the dopamine neurons express the dopamine transporter. None of the other neurons express the dopamine transporter. And actually, this is important for the treatment of Parkinson's because, as many of you know, we often use L-Dopa or other types of dopamine-modifying drugs to boost the level of dopamine in the extracellular space. Because when there's too little dopamine caused by the loss of the dopamine neurons, this of course causes the main symptoms of Parkinson's, rigidity, lack of movement. So we want to boost dopamine in the extracellular space, and that is very efficiently done, for instance, through L-Dopa. What L-Dopa does is it actually hijacks non-dopaminergic neurons, and then it causes them to release dopamine. So that's good. You get more dopamine in the extracellular space. But now you're lacking those adult dopamine neurons that can clean up the dopamine. And this is a problem because this means that you can sometimes get too much dopamine in the extracellular space, and this causes involuntary movements, or what we also know as L-Dopa-induced dyskinesias. And this is actually one of the main reasons why when the dopamine axonal terminals, the dopamine neurons, are progressively lost in the disease, it also means that the dopamine-modifying therapies are losing some of their efficacy, and you get more and more therapeutic medication. And if you imagine this is like how much medication you have in the blood, it works very well. And when you're in the white window, you're kind of in a good, you're in good symptomatic treatment. And when you get into the pink window here, you feel the side effects of the treatment, typically seen as dyskinesias. So this is something that can be an issue for many individuals living with Parkinson's disease. It can sometimes be very severe. This is a video from Parkinson's UK that I borrowed, showing just how severe it can be with these L-Dopa-induced dyskinesias. So we wanted to test if our stem cell-derived, our lab-grown dopamine neurons, when they grow up and mature in the brain of the rat, Can they now clean up after themselves? Can they remove the dopamine if there's too much? And we would want to test this to see if they can actually ameliorate these L-DOPA-induced dyskinesias. So we collaborated with Emma Lane, who's at Cardiff University, and she has this rat model, which she first lesions the dopamine neurons, and then she gives the rats L-DOPA every single day, and she does this to induce these L-DOPA-like induced dyskinesias. We also call them abnormal involuntary movements in the rat. And you can see this here, kind of the rat is doing this excessive grooming. It also starts rotating around itself. And we then asked, okay, if we now transplant stem cells into this rat, we continue to give it L-DOPA, so it should continue to have dyskinesias. Can we now lower these dyskinesias? And in fact, we saw that we could, over a period of around 15 weeks, these dyskinesias were actually completely gone in these rats. And this, for us, is a sign that the transplanted stem cell-derived dopamine neurons can not only produce and release dopamine, but can also reuptake dopamine if there's too much. And that's super important to maintain the correct level of dopamine in the extracellular space. So with all this very nice data, we thought, we want to take this into humans. We want to see if this can also help individuals with Parkinson's disease. And I'm actually very proud to be in Edinburgh today because this whole journey actually started in Edinburgh. We use a cell line which is called RC17, which was derived at Roslin Cells, which is located here in Edinburgh. And this is actually the source, we call it the source cell line of our stem cell product. So 2 vials, very precious vials of the RC17 line, were shipped from Edinburgh to London. And in London, we did what we call clean room manufacturing, which means that there are some people wearing these very special suits, super sterile, super controlled conditions, because now these stem cells are intended to be put into humans and not just into rats. So of course the standard is much higher. And this is what the manufacturing looks like. This is our dopamine teenager progenitor cells, the immature cells that are growing here in the flasks, and which were then frozen down in a cryotank in London and shipped to Lund University. And with all, we then wrote up a lot of data on these cells, all the tests that we've been doing, all the animal experiments, all the experiments we've done in the lab. To get permission to go into humans, you have to write all this up in a huge document package, and you send it to the regulatory authorities. In the UK, that would be the MHRA. In Sweden, it's called the MPA, and we actually sent it to both of these authorities. And after 1 year of questions back and forth and a lot of answers and new questions, we finally got permission to go into clinical trial. And on February 2023, we transplanted the first patient in Lund. And we have now completed transplantations in this trial. I unfortunately cannot yet show you data because we're still in the follow-up phase from this trial, but I'm going to show you some data from some other trials in a little bit. And the reason is that these trials are very long. First of all, it took us 2 years to just transplant 8 patients in the trial, and that's because you have to stagger and wait to see that it's safe between each individual. And then once the participants are transplanted, then there is a long follow-up, actually up to 3 years. And this is what, just so you know, you can relate to what it means to be part of a stem cell trial. We demand a lot from our participants. They need to come for MRI scanning several times during the first 6 months. And then they do PET scanning, fluorodopa, P2I, and then of course they're assessed for motoric function through the UPDRS test. And of course the most difficult one is when we ask our participants to go off medication for actually 48 hours. They have to go off medication to do the UPDRS off testing. And this is very important for us to be able to assess the effect of the transplant. And we are, of course, very grateful to the participants in our trial because we know we ask a lot from them. And some of you may be familiar with this guy, Andy Cassie. He's one of our participants. And also, I wish I could say that the graft made him a very good ping pong player, but I think he was a good ping pong player also before he was part of our trial. He writes some blog posts also about his experiences of being part of the trial. So if you're interested, you can also follow that. So now that I disappointed you with not being able to show data from our own trial, I'm going to show you what else is going on worldwide. Because we are, luckily, not the only ones working on this. And I think that's very good, because we need to see what works best. How do we compare between products from different groups? And this is now the status of clinical cell products that are in dopamine cell products that are in clinical trial for Parkinson's right now. We had the first trial that was initiated was in Japan, in Kyoto, the Saira Institute. They transplanted 7 patients. Then there was the Blue Rock trial in US and Canada. They had 12 participants. STEM-PD trial, which is our trial, with participants from Sweden and UK, in total 8. A trial in China, which unfortunately we don't know very much about, so I don't really have any information on that. They say they have transplanted 50 patients. A South Korean trial with 12 patients. Another Chinese trial with a site in the US. They say they will transplant 46, but we haven't again seen any data from this. And then I just want to briefly mention to you, I told you before about this way that you can make pluripotent stem cells from a fertilized egg, and that's what we did in our case. We used the RC17 cell line, which was made in this way from a fertilized egg, but now you can also make pluripotent stem cells to another route, which we call reprogramming, and that was invented by this very brilliant guy called Shinya Yamanaka, who has also been to Edinburgh to visit Thilo and the Stem Cell Center. He won the Nobel Prize for this technology, and what you can do now, because of his technology, is you can make stem cells from each individual. So from each one of you, we could take a blood sample or a skin sample and make that pluripotent stem cell from you, which means that we can now make your neurons, your brain cells. We could also make your own kidney or your own cardiomyocytes to repair your heart. So you can see there's a lot of potential in this. And this technology now means that we have 2 different routes to make stem cell therapies. One is where you get a donor tissue. That's what we've done. You get a stem cell that comes from another donor. But through this reprogramming technology, you can also do what we call autologous transplants, which means that you can make stem cells from each individual participant and then make those into neurons. So neurons that belong actually to the individuals themselves. There are some pros and cons. For this approach, you need immunosuppression. But for the brain, only around 12 months. For this autologous approach, you, in theory, don't need immunosuppression, but it is super, super expensive. It's very expensive to do autologous transplants. You need to make basically a new product for every single individual, and that is very challenging, and I'm not sure this will ever be viable actually on the market. But nonetheless, there are groups that are working on this, and we actually now have 3 trials with autologous cells, meaning trials where the cells have been produced from the individuals themselves. One company called Aspen Neuroscience in the US, they have already, well, I'm not sure actually, they are going to transplant 9 individuals. I think they've transplanted 4 so far. At Harvard, there is also a trial with 8 individuals, and another one at Harvard with 6 individuals. So this is what is ongoing at the moment. 2 of these trials have published their results. I'm just going to show you what that looks like. It's the Blue Rock trial in US, Canada, and it's the Saira trial from Japan. So this is the UPDRS data from the Blue Rock trial. At 18 months, they saw around a 25% decrease in UPDRS off score over 18 months. This was 5 individuals. This was with the low dose. Now they also had a higher dose, which was 3 times the amount of cells. And here they actually saw a 50% reduction. You can see they went from 49 UPDRS score to 25 over 18 months, which is quite significant in the off state. So this data was quite convincing, and actually they showed also their PET results. There was a little bit, we think the PET results are not yet showing such a significant effect on the dopamine, but they will soon be publishing. We know also the 24 and 36 months follow-up, so we're looking forward to that. What is quite interesting about this trial is that they have announced that they have been given permission to go from their first clinical trial, which is what we normally call phase 1, and go directly into phase 3, which is the last clinical trial. Normally, you have to do phase 1, 2, 3. That's the normal route. But FDA gave them permission this year to skip phase 2 and go directly into phase 3. And they just released, actually around a week ago, this press release saying that they've now started their phase 3. They transplanted the first individual in their phase 3 trial. So this is actually the first example we have at all in the world of a stem cell therapy that has gone into a phase 3 trial. This is very exciting. They are going to transplant, I think, around 100 individuals in this trial in Toronto and New York, and maybe they will also have a European site. We don't know that yet. The Kyoto trial showed some variability between patients. There were some patients that did very well, others that didn't show much effect, so a bit also like we've seen from the Fetal trials. They had less patients, they had only 7. We saw individuals with very prominent effects on the PET scanning, which really showed us that the dopamine, the transplanted dopamine neurons were surviving, and this was also an individual that responded very well to the therapy. And the Kyoto trial has now, the product has now been taken over by a company called Sumitomo Pharma. And they also just actually 2 months ago released this press release saying that they are now going to apply for marketing authorization in Japan. And this is kind of a little bit special because in Japan they made a special rule that when you have a stem cell therapy, you can just do 1 clinical trial and then you can start selling your product on the market if you've shown some efficacy from the product. So actually, this dopamine product is now going to be sold on, very likely going to be sold on the Japanese market during this year. They are obliged to continue to collect data, but this is a very special case which is only allowed in Japan. In parallel, they're also starting a phase 2 trial, 2 phase 2 trials in the US to enter the US market. So, okay, sorry about that. So in summary, we've seen several fetal tissue trials that have been completed. I'm just going to summarize what I've been telling you today. Some patients have shown remarkable improvements, but there's been a lot of variability in these trials. Right now, we have 9 clinical trials initiated with human pluripotent stem cells. 3 of these are autologous, meaning it's the patient's own cells. 6 of them are with donor cells, or allogeneic cells, as we call them. 2 clinical trials have been completed. Blue Rock with 12 participants, Kyoto with 7. This is the data I just showed you. 1 phase 3 trial has been initiated. That's the Blue Rock Therapeutics clinical trial. This is the first time ever we have a pluripotent stem cell product that is in phase 3, so that in itself is really very remarkable that Parkinson's is really leading the field here. It will be very interesting to follow their data during the coming year. And sale on market will be initiated in Japan by Sumitomo. We will also have our data coming out likely early next year from the STEM-PD trial. Lastly, before I round off, I just want to inform you that you may find stem cell treatments offered by private clinics around the world. That also happens here in several clinics in Europe. I just want you to be mindful that not all, well, most of these stem cell therapies that are offered by private clinics are unproven, so please be vigilant. You're very welcome to write me an email if you stumble across something and you're like, what is this? is this true? Should I do this? Should I not? Please do so. We do have problems with clinics that are offering therapies that are really not working, but they are still convincing individuals to come and pay a lot of money for them. So just be vigilant about that. So lastly, I just want to say that this is, of course, not me doing all this work. This has been an effort by a lot of people, a lot of EU consortia in which we've worked together, several hundreds of people, and the STEM PD team, which has really been led by Malin and Roger and myself, together with our very important partner at Novo Nordisk, who has been our partner and are now they have taken over the development of the product, so they will be the ones to run phase 2 and phase 3. We have the neurologists, Gesine and Håkan from Lund, Hjalmar, who is the neurosurgeon, and of course all the clinical trial teams in Lund and Cambridge. And most importantly, I want to thank the participants of the STEM-PD trial, because without them, of course, our research would not be progressing. And thank you for listening.
I'd just like to ask you a question. So Agnete has given us a great talk, and she's kept to time. Before we have the question and answer session, we can have a short break, for a comfort break for about 7 or 8 minutes. But I'm going to ask Agnete one question before we all go to the loo. Your cooking skills, growth factors, and getting the right recipe, it's amazing. How are you in the kitchen with your cooking?
I better leave that to others.
Okay.
Welcome back. We're going to get started with some Q&A. There are 2 roving microphones, one over here and Noelia over there. And also in the middle area, there are some microphones like this. You just need to press the buttons for us to hear you. And also the people online. Yeah. So and I have maybe 30 questions online that we're not going to get through, but I really want to start for live questions for Agnete. Does anyone have some questions that I thought? Oh, yeah, sure. Let's start with you, sir.
Thanks very much. That was great. What's the chance of it helping me or us?
Yeah. So you're asking what's the chance of being part of the trial? Yeah, and I wish I could say we could include all of you. Our own trial is ended. We already included the participants. That was only 8. Now we've handed it over to Novo Nordisk, which is a pharmaceutical company. They will be running phase 2 and phase 3. And the current plan is there will be a site in the UK, and a site in Sweden, and maybe also in other places. So I very much hope that trial will be up and running next year. It's something you can check out on the Novo Nordisk website, because they are now in control of the trial. Of course, still it will be a limited number of participants, and it also there is also, of course, eligibility criteria, but that's where you can keep yourself updated.
That's a very good question, very clear, obvious question. Yeah, over here, Steve.
Thanks. A very interesting and stimulating presentation. So thank you very, very much for that. I guess when I look at the procedure, it's going to be limited by the number of surgeons who can actually do the procedure.
Yeah.
Is there any research going on to identify if there's a different drug delivery mechanism to get whatever is required to the right part of the brain without using surgery?
I mean, this is really the bottleneck of the stem cell therapies, is that there is no magical way, unfortunately, to get a neuron into the brain without placing it there. These neurons, they don't migrate, and that's because they're not supposed to migrate. If they were migrating, there would be something wrong with them, actually. So it is a requirement that we put them in the brain exactly where they're supposed to be. In this case, we place them in the putamen. And you're absolutely right, that is a bottleneck of the therapy. We do use, the surgeons we use are very skilled DBS surgeons. And it's very much the same procedure as the DBS surgery. So we do believe that to scale it out, it would be the same surgeons as for DBS. And of course, it's only a single surgery, and then hopefully it works lifelong. But you're absolutely right, that is a bottleneck. Yeah.
I think it's a good point that the expertise exists, so the DBS surgeons can do it, but
Yeah.
you're right, the number of them is
Yeah,
fixed.
It is, yeah.
Capacity for DBS surgery in Scotland is very, very low.
Yeah, it is a problem, yeah. Well, maybe this can get us to educate some more surgeons, yeah.
Yeah. Okay, up above.
Hiya, thank you so much for your talk. I'm a medical student here at the university, and I just had a question about the types of stem cells. So you talked about the difference between the allogenic approach and the autologous approach,
Yeah.
but for the allogenic trials on your summary slide, some of them use the human-induced pluripotent cells and some of them use the embryogenic cells.
Yeah.
So which one, is there pros and cons associated with that as well, or?
Yes, so we use embryonic stem cells, and that's because it is the least manipulated cell. I mean, the iPS cell, the induced pluripotent stem cell, goes through this additional genetic manipulation, which we personally wanted to avoid just because it can cause some extra genetic abnormalities. And you are also right that there is scientifically actually no reason to use an iPS cell if you're doing allogeneic transplant. Nonetheless, many companies are doing it, and it's mostly, I believe, for political reasons, because there are countries where embryonic stem cells are not easy to work with, and they are worried about repercussions in working with embryonic stem cells. So these decisions are actually very political in the end, unfortunately.
I think Marato from Dundee. Yeah.
First of all, thank you very much for that talk. That was stunningly clear, and thank you for sharing that. You mentioned early on about the variability in the original trial and how that may have been related to how the cells were being isolated from the fetal midbrains. Were you surprised by the variability that's now being seen with the two trials that's reported? Can we learn anything about the variability from the data they've shared about which patients are perhaps more suited to this approach?
Yes, I mean, we were hoping there would be less variability because now the product is much more standardized, right? And basically all individuals are receiving the same product. I think this highlights to us that there are challenges. We were discussing this before. That it is a challenge to get the cells to survive at this time point where you put them in the brain. It's very stressful for them. And the environment they come into can be different between different individuals. And unfortunately, that's something we can't control, and we also cannot see it, right? We can't see what the environment looks like before we transplant the cells. Maybe we should be better at trying to investigate that. But this is, I think, the current challenge is to get the cells to survive equally in every single transplanted individuals. That will be crucial. And this is the kind of thing we only learn by going through the clinical trial, because in a rat, it's pretty easy, right? They seem to survive all the time, but the rat is, of course, not a human brain. And there are tricks to do this. I think we just need to work with them.
Yeah. So for the online audience, if you want to upvote the most popular questions, please do. But the most popular one I'm going to ask now, if that's okay. So is it possible that stem cell therapy will be effective across a wide range of symptoms, or will it be just limited to motor symptoms?
Yeah, so it's important to say that when we put a dopamine neuron into the brain, it is intended to alleviate symptoms that are connected to dopaminergic deficiency. So that means symptoms that you would typically respond to with dopaminergic medication, but in this case would be with less side effects because you don't have this systemic release of dopamine. It's only produced where you need it, and you don't have too much. Like I showed you, it can reuptake. We do not expect that the dopamine neurons would alleviate symptoms that are related to other brain regions, such as cortical, cognitive effects, for instance. There has been some reports in some of the fetal trials that it may help on depression symptoms, and that may actually be a secondary effect, right? Because if your sleep is very poor and you get a dopaminergic transplant, this can maybe improve your sleep quality, and that in itself, of course, can have secondary effects on mental health. So there may be some secondary effects of that sort, but basically it is designed to alleviate dopaminergic symptoms.
one more online. I think this is a difficult one, so we'll give you a hard one. What's the worst outcome of the stem cell therapy? If it doesn't work, can it harm function?
Yes, so of course there's always risks of every single trial and every single treatment, and of course there also is of this therapy. One risk is of course the surgery itself. We have an inclusion criteria of up to 75 years of age for inclusion in the trial, and that was primarily to not have too high risk in the surgical procedure. And then there is the immunosuppression, and we actually, unfortunately, in our trial, we had a participant who got a very severe infection during the trial and succumbed from that. It was a very severe fungal lung infection. And of course, infections like that, it's not caused by the transplant, but the risk increases because the individuals are in immunosuppressive treatment. So this is, of course, something we discuss a lot, how much immunosuppressive treatment is needed, and maybe we don't need as much as we had in our trial. So this is something we're learning as we go along. One thing that was very positive, I think, we've seen from these trials is that in previous fetal tissue trials, there was what's called graft-induced dyskinesias, which means that some of the individuals got dyskinesias from the graft, so not L-DOPA-induced, they were independent of L-DOPA. And we later learned that this was because of contaminating neuron in the graft, and we are now very good at getting rid of that subtype of neuron. We don't have those in the stem cell transplants, and also in all the trials we've seen so far, there have not been any graft-induced dyskinesias.
Yeah, that's great news. That's...
Hi, you mentioned some remarkable reductions in new PDRS scores. Are they all similar scores but at the start of the trial, or is there a range of them?
Sorry, can you repeat?
Have all the patients got similar scores pre-trial?
If they get scored before the trial?
Yeah, are the scores all very high? Are they mid-numbers before? You mentioned some have 50% reduction.
How high were the scores? So is that your question?
Yes, how high were they before the trial?
So in the Blue Rock trial, they were quite advanced. I think the scores was between around 50 on average for the participants in the Blue Rock. Yeah, actually they were. They went from 49 to 25 in the off. This is part 3 in the UPDRS. They went from 49 to 25. So they were quite advanced in that trial. In our trial, we have, they're less advanced. So they're in the category of what we would call moderate Parkinson's.
If I can jump in, let's go back here first.
Yeah, thanks again for the talk. Some of these stem cell trials we see will have a placebo arm or a sham surgery arm. What's your thoughts on the scientific value of that, and also, I guess, the ethical side of it?
The ethical side of the stem cells?
Of having a sham surgery arm or a placebo.
That's a super good question. Yeah, so the question is, how do you do a trial with a placebo-controlled arm? This is what you would normally have to do in a phase 3 trial. And of course, the big complication when you're working with a stem cell treatment is that your control arm now have to believe that they have received the stem cell transplant. So they have to go through anesthesia, a sham surgery, which means they're on the operating table. They get a burr hole actually in their skull, but nothing is injected into their brain. They lie there for the 8 hours or so that the procedure takes. They wake up, they get immunosuppressive treatment because otherwise they would know that they are not in that they're in the placebo arm. So there's a lot of ethical complications around that. We've discussed that over and over again. It's a very good question. The FDA still is very adamant. They require sham surgery control. So the Blue Rock trial will be with the sham surgery arm. They will receive immunosuppressive treatment. In Japan, for instance, they find this unethical. In Sweden, they find this unethical. So trials in different countries will actually have to be designed differently. And we're still discussing how to solve that.
Yeah, what's your thoughts on the scientific value of it?
Of the sham surgery? Of course, it's a classical control, right? And of course, it is the way to alleviate placebo effects. But it is a very strong ethical question whether you can actually, is it ethical to do that? Roger Barker has very strong opinions about this. His idea is you can make a very large control group that you follow continuously over many years, because then you get a very stable data set for your control group, and that can be a way to get around having a sham surgery control.
Yeah, it's a tough one.
It's a tough
but,
one, yeah.
you know, the video you showed of the MPTP patient, you know, that type of recovery, if we do see something like that, it can't be placebo.
Exactly. If the effect is strong enough, then I don't think you have to have placebo.
This is, I think, a question that might be relevant to many people here. This is a simple one. What are the minimum qualifications to participate in one of these trials?
You don't need any qualifications.
Well, criteria, I think inclusion criteria. I mean, but they wrote qualifications.
In our trial, it is moderate. It's classified as moderate Parkinson's. You have to be responsive to dopaminergic medication because otherwise you probably won't respond to the graft. You have to be maximal 75 years old, and you have to have had Parkinson's for at least 10 years. That was for phase 1, I will say. That will change for phase 2 and phase 3. It's always a very confined group for phase 1, but it will change as we go along.
Any more questions from the audience? Or should I? Oh.
Just a quick one, Charles. The Americans, the FDA, allowed the trials to jump straight from phase 1 to phase 3. Why would they allow that where elsewhere it has to go phase 2, phase 3?
Yeah. So when I talk to them, what they say is that this is something that also happens in the gene therapy field and for orphan diseases, so rare diseases, because it's a way to get treatments faster to patients. And I think what they have argued in this case is that even though Parkinson's disease is not an orphan disease, it's not a rare disease, the treatment is not a treatment for all Parkinson's patients, right? It is for a subtype. It's something we see as a treatment for, in particular, young onset, because they will have to live with the disease for many years, right? So they are the ones that will benefit mostly from it. And I think this is the argument they had put forward, that it's kind of an orphan treatment, even though it's a more common disease.
It's nothing to do with the fact that the American one is based on cells from your own body, whereas the other ones are cells from
the Blue Rock
donors.
trial is not autologous.
Right, okay.
It's not based on cells from, it's also embryonic stem cells. Yeah. Exactly. That's very true. My worry is if you make an autologous transplant, only Elon Musk can afford it.
Okay, some of the questions are not anonymous. So this one is from Professor Dario Alessi in Dundee. Do you have a current estimate for the cost of treating a single patient? And how realistic would it be to scale this approach to the roughly 10 million people living with Parkinson's?
Yes. So actually, I mean, this sometimes keeps me awake at night because I won't be the one to set the price on a final therapy, right? And we have tried to really make the production of the stem cells very efficient. So actually, the manufacturing costs themselves are not that high. It's around 5,000 euros per patient. So that's not too much. But, of course, this will not be the market price because the market price is actually not, unfortunately, not based on manufacturing. It's based on value. So when the pharmaceutical companies finally get a product on their market, they don't ask how much did this cost to produce. Instead, they ask how much money are people willing to pay for it. And that, of course, means that the price can be much higher. That's also a reason I'm very happy we have many competitors, because only with a lot of competitors on the market will the price go down.
Yeah, that's an excellent point. Yeah, the amount of competition in this area is much higher than I thought it was going to be.
It's very good.
But it is good for the price.
It's very good.
It should bring the price down. Yeah. Oh, there's a question here. Yeah. Oh, no, I can go online, yeah?
Yeah.
What criteria has to be satisfied for a stem cell treatment to be suitable? I'm not sure what that question means. What criteria? Maybe that was a similar question to the inclusion criteria. So this a common question I've also seen is, what is an exclusion criteria?
An exclusion criteria, yes. So in our case, it's if you're above 75, it's if your Parkinson's is very advanced, or if you're Hernan-Jarre stage 4 or 5. It's also comorbidities, and it sounds harsh that we can't take individuals that have other diseases like type 2 diabetes or dementia or something else. And unfortunately, that's just because when you design a clinical trial, you want a very clear population of participants. So you tend to try to make them as homogeneous as possible to get a better chance of seeing a signal in your treatment.
What if you?
That's of course because you've already had a brain surgery and because it will be a confounding factor in the motor assessment.
Okay. Oh, at the very top.
I had a question about how might combining stem cells with gene editing, for example, CRISPR, help correct mutations linked with familial Parkinson's disease?
Yeah. So you mean to correct in the stem cells?
Yes.
Yeah. You can do that. So you could take a stem cell from an individual with a genetic mutation, a familial mutation, and you could correct it with a tool that we call CRISPR, and then you could put it back. Again, it would be super expensive, very expensive. So I don't think we're ever going to see that on the market. I think we're going to see allogeneic treatments. One thing I didn't mention to you is there is actually this very cool technology that's coming up, which is what we call the stealth technology. You maybe know these fighter jets that have this stealth technology. It means that they kind of fly under the radar. The radars can't see them, right? And we have something similar in stem cell biology, which is that we can make stem cells that fly under the immune system radar. So they actually become invisible to the immune system. And actually, we're very exciting times now because just a few weeks ago, there was a press release out from an American company that transplanted one individual in Sweden, a type 1 diabetic person, with stem cells with this stealth technology. And they've now shown that 6 months after transplantation, even though this individual received cells from another donor that wasn't matched, the patient has been without any immunosuppression, right? So just received the cells. The cells are still alive 6 months later. That is super impressive, and it's really a game changer for producing these stem cells because it means eventually, hopefully, we can completely get rid of the immunosuppression. And then we don't need the autologous technology at all.
Yeah, that's a really good question, really exciting news. Online, this is a good one. Do transplanted dopamine neurons suffer the same process of degradation as the patient's original neurons?
Yeah, so the question is when we put new dopamine neurons in, do they also get the pathology that we see in the Parkinsonian brain? And actually, we have learned, because we now have some post-mortem brains from these fetal tissue transplant trials, we have actually been able to look at them many years, even more than 20 years after transplantation. And yes, indeed, some of the neurons in those transplants have also acquired Parkinson's disease pathology, in particular this alpha-synuclein aggregation, which Thilo studies a lot, has entered into the transplanted neurons. But very slowly, it seems around 0.5, when they quantified around 0.5% of the neurons per year become affected by Parkinson's pathology. So very few, and around 20 years, we would expect around 10% are affected. So we believe the majority of the neurons in the graft will still be functional for many years.
When I ask the question to Blue Arc, they say the same thing, that it's very, very, very slow.
Yeah.
Oh, yes, Allison over here.
I wanted to ask you whether there's any influence of gender in the work that you're doing, whether gender is taken into account, and if not, why not, and if it is, in what way?
So of course, we're always required both when we do the preclinical studies in the animals, but also in the clinical trial, to ensure a certain gender balance. So we're not just testing this therapy in one gender. Of course, in Parkinson's, there is a bias, right? So we do have an overweight of males in our participants, which is reflective of the general population of Parkinson's individuals. And this is what trials will normally look like this. So you have a balance which is reflecting the patient population. we don't have enough patients to be able to make any statistical conclusions between males and females in the study. It's only 8 individuals. But of course, once you reach phase 3, the larger trial, you have to be able to make statistical conclusions if there are any differences. We are not expecting any differences because it's a very local environment in the brain. We don't see any reason why it should differ between men and women.
Yeah, it's a very important question. So as she's walking, I'm going to ask a quick one from online. Will this therapy be effective for people with genetic Parkinson's, inherited form?
Yes, absolutely. And actually, some of the genetic forms we think are very amenable because they are very clean in their dopaminergic loss and don't affect so many other regions of the brain, some of the LRRK2, for instance. So we think that absolutely, yes.
Great. Okay, I think one more question we can do. Yeah, one more, please.
Yeah, thank you very much for your talk. I'm wondering, how is the distribution of the stem cells in the brain assessed?
Yes, so the only imaging technique we have available is PET imaging. So where we can look at tracers that bind specifically to the dopamine neurons, which I showed you in the pig also. This is the only way that we can see the graft, but we can then only see it once it has become finally mature. In the beginning, it's very difficult. It's a very small graft. So we can see it a bit in the beginning by MRI, and then once it's matured by PET scanning. But it is difficult. Yeah.
Brilliant, thank you. Agnete has offered to answer some more questions online that we'll hopefully put on the website afterwards. But join me in thanking Agnete for a brilliant lecture. Yeah. Okay, so one more set of remarks, and I'm really pleased to introduce Martin Taylor, who has a young onset Parkinson's and is a massive Hearts fan. Oh my God.
First test. Just water, please. Okay. Right, here we go. Firstly, thanks to Agnete for her brilliant, inspiring, intriguing presentation on what's long been one of the most hopeful and sought-after therapies in development for those of us living with a Parkinson's diagnosis. I recall watching videos on YouTube dated as far back as 1991, featuring the former Arsenal and Liverpool footballer Ray Kennedy, who was diagnosed with PD age 32 in the early 1980s, which referenced cell replacement therapy as something to be hopeful for. Similar to Michael J. Fox, when he first went public with his diagnosis in the late 90s, it was also something he placed great hope in. This illustrates both the enduring hope, but many frustrations that have arisen, to both those patients with hope of a better life and clinicians and scientists who passionately believe that cell replacement therapy could be the way to deliver it to them. When David asked me to do the closing remarks this year's Edinburgh Parkinson's lecture, my immediate reaction was to say yes. Although I have not been personally involved in this particular study, I have participated in many others. I know at close hand how passionate, determined and motivated that many of the researchers are to achieving better outcomes for those impacted by this horrible disease and their families. In particular, our own Teal Cunath, whose work on stem cells and PD and many other projects has been a source of inspiration and hope to many in the Edinburgh PD community and beyond. Water. A while back, I co-authored an academic paper. Well, to be more accurate, Teal authored it, allowed me to be the honour of being listed as a co-author, in which it was suggested that patients carrying a mutation in the Parkin gene, of which I am a carrier of a heterozygous variant, should perhaps be priority candidates for cell replacement therapy, for a variety of reasons, including the physical symptoms being slow, progressive, mild, don't feel very mild at the moment, to be honest, a lower instance of cognitive impact, We are now beginning to see the recruitment for trials based on this premise in the US. I would also like to acknowledge the bravery of any patient who willingly puts themselves forward to participate in clinical research, especially those that may involve sham surgery as part of the placebo, as is required for regulatory approval in some countries. Clinical research is dependent on the courage of these people and their willingness to put themselves through what can often be a traumatic ordeal to help advance the knowledge to help others. Having lived with PD for 11 years now, having been diagnosed aged only 32 in 2014, I've been hopeful that through studies like this, there may be a better tomorrow for me and my family, and others and their families too. Thank you.
So it's time for a vote of thanks. Thanks to local company Roslin CT for sponsoring the reception. I think you'll understand why I offered them the opportunity to do that from Agnete's talk. I'm very pleased that they agreed. So thanks to Roslin CT. Also, thanks to Lindsay for the welcome. Thilo for his, as usual, expert introduction and Q&A, which is beyond me. Thanks to James Jopling and the Parkinson's UK Scotland staff for all the help they give me to run the lecture. And also thanks to the Edinburgh branch for paying for most of it. And to the RCPE team for their excellent putting on of a good show. And thanks to Agnete for giving us hope that an effective therapy for Parkinson's isn't that far away. Most of all, thanks to everyone who's here today at the Royal College for watching, and for those who are watching online. I think we've had a really good evening, and I hope you think so too. And before you go, we could do with some money to sponsor the work of Parkinson's UK to find a cure for Parkinson's. As you leave, there'll be people with a bucket close to either entrance. You're happy to chuck some notes into that. Doesn't matter if you haven't got any money. We cash or card accepted, so you can use your card as well. And for those of you who are watching at home, you can also donate online by going to the Just Giving link, which you can see on the slide at the top. And once you're on the Parkinson's branch Just Giving page, if you type Parkinson's research in to the message box, I'll make sure that all of that money goes for Parkinson's research. Thanks to everyone for watching, and for those of you in the audience here at the Royal College, safe trip home. Thank you.